Hello, everybody. Welcome back. So now we're on to the fun part of chapter 10. I think it's the fun part. It's the physiology of muscle fiber contraction. So this is where all of the anatomy and the parts and pieces that you just spent a couple mini lessons learning about is going to actually start interacting to do something, to do a contraction of your muscle fiber. So there's actually a couple more physiology videos. I have them broken down into physiology of the muscle fiber, physiology of the whole muscle, and ATP production. So those are kind of the three major concepts of physiology in chapter 10. So muscle fiber contraction, whole muscle contraction, and ATP utilization. So keep that in mind if you're kind of consolidating or compartmentalizing your information of chapter 10, because I know it's a big chapter. So in the notes, after the end of the anatomy of kind of the sarcomere and the thick and thin filaments, there's a thing that says the sliding filament theory. And so the sliding filament theory isn't really a theory in like a guess kind of thing. So remember, scientific theories are actually things that are supported by lots of evidence to explain a natural observation or phenomenon. So the sliding filament theory is the idea that muscle contraction occurs because of the filaments, the thick and the thin filaments are sliding over each other. So thick and thin interact to contract and relax, and contract and relax. So this is what we have to make all of those parts and pieces that we just learned work into doing that. Okay, so this is our explanation of the sliding filament theory. The name that we now usually call it is the excitation contraction coupling. So we have to excite the skeletal muscle tissue, and then we have to couple that excitation with the actual contraction of the muscle tissue, the skeletal muscle fiber. So we're going to first start taking a look at the excitation part, which is the arriving nerve impulse to a muscle fiber. Okay, so the kind of overview picture that we're going to start with is this one right above, like right by my head here. So imagine you have a motor neuron, probably located in your spinal cord if it's, controlling your arms and legs, or I guess in your brain if it's controlling your face. So you have a motor neuron, and its job is to stimulate a muscle cell. So you have the neuron, it runs its signal all the way down the length of the axon, and then communicates to the muscle cell at the sarcolemma. Okay, so remember, sarcolemma is the plasma membrane of the cell. At this very specific location of where the neuron interacts with the sarcolemma, collectively this is called the neuromuscular junction. So that's why I titled this slide the neuromuscular junction. Here you can actually see a micrograph of the neuromuscular junction right here. So here's the muscle fiber, and here's the neuron fiber. And so that's where we're going to see the arrival of the nerve impulse, what we call an action potential, going to be released here at the sarcolemma. And then at that location where we have the receptors to pick up the neurotransmitter, this is called the motor end plate. And what's unique about the motor end plate is it's wrinkly, right? So it's not a nice smooth sarcolemma like we saw in all the other pictures. It actually curves in. So here's a micrograph showing the motor end plate. Okay. And then on this side, this is the motor neuron. So that's part of the neuromuscular junction. And every one of those tiny little circles that we see in the micrograph are representative of the vesicles. So these are vesicles. They're little transport pouches, if you will, that are inside of the neurons. And inside of the vesicles, we have what's called a neurotransmitter. And the neurotransmitter that we are going to be focusing on here is called acetylcholine esterase, or acetylcholine. There we go. Let me find the right one. Acetylcholine, ACH. We're going to talk about acetylcholine esterase in a little bit. So acetylcholine is the neurotransmitter sitting inside of these vesicles, hanging out at this very end part of the motor neuron, right a little bit of distance. The synapse is the distance between the end of the motor neuron and this motor end plate, which is the folded little regions of the sarcolemma. All of this together is called the neuromuscular junction. Okay, so what we're going to see is when the action potential arrives, which is right here, action potential arrives kind of that reddish lightning bolt. The arrival of the action potential causes these vesicles to exocytose, right? So they are phospholipid bilayer. They fuse with the phospholipid bilayer of the membrane of the neuron, and they release their neurotransmitter into the synaptic cleft. Okay, so we've just released millions and millions of molecules of acetylcholine into the cleft. Now, embedded onto or in this membrane are the receptors. Okay, these little purple cube-looking things. So this is a chemically gated channel. So if you remember chemically gated and G proteins and enzyme linked receptors, this is a chemically gated or an ion channel linked receptor. So the acetylcholine is the signaling molecule. It binds to the receptor. When that receptor binds, it opens an ion channel, which allows sodium to rush in. So we have this huge influx of sodium, and what that does is it triggers an action potential on the sarcolemma. So it's almost like a game of like pass the baton, where the baton is the action potential. The neuron has a signal, it crosses the synaptic cleft, handing it to the next cell, which is the muscle cell, and then the action potential, the signal then gets traveled along the sarcolemma of this stimulated muscle fiber. Okay, so here we see the action potential is now traveling along the sarcolemma. Now, in a neuromuscular junction, in this synapse, we do not want the acetylcholine to continue to stimulate the cell. Otherwise, our muscles would stay contracted and we wouldn't never have any relaxation. So this is where that ACHE comes in. This is an enzyme, acetylcholine esterase. It's those little yellow crescent moon looking things. What they do is they break down the acetylcholine. They remove it from that binding site. The gates close. No more sodium is flooding into the cell, and the stimulus is done. So the muscle gets that one stimulus, it shoots off its action potential down the rest of the muscle fiber, and then the neuron is done firing its signal. So these are the events of the neuromuscular junction. So the arrival of the action potential, exocytosis of the neurotransmitter, binding to the receptor, opening those sodium channels, sodium rushes in, excites, right? Here's our excitation, excites the sarcolemma, and then acetylcholine esterase comes in, or is always there, but is starting to break down the acetylcholine, stopping the signal on the muscle. Okay, so this is the excitation part of the coupling, all right? So make sure you're comfortable with those steps. So here I have a little GIF that's kind of running up in the corner, so hopefully that's not too distracting for you. So here's what we just did, right? So here's the excitation part of the contraction coupling piece. So the action potential runs down the neuron, we release the acetylcholine, we allow the sodium to rush in, and now we have this excitation. Here we have our action potential, and if you remember anatomically, the T tubules are continuous with the sarcolemma. So any electrical activity that's happening on the sarcolemma will get converted and carried down deep into the cell via the T tubules. Okay? Now again, you got to remember, this is where all that anatomy knowledge is going to come in. Because if you remember, the T tubules were right next to the terminal cisternae of the sarcoplasmic reticulum. So here's a triad. Let me get a different color. So here's a triad right here. Okay, so two terminal cisternae and a T tubule right in the middle. So when that excitation, when that action potential comes down the T tubules and comes right next to the terminal cisternae of the sarcoplasmic reticulum, it triggers this release of calcium, these calcium ions. Calcium inside the sarcoplasmic reticulum is highly concentrated. So when this signal shows up at the sarcoplasmic reticulum, ion channels open, specific for calcium, and so now the membrane is permeable to calcium ions. Think back to general bio. Which direction do things like to go? From high concentration to low concentration. So we have high concentration in the sarcoplasmic reticulum, low concentrations in the cytoplasm or sarcoplasm. And so when those channels open, calcium just floods out of the sarcoplasmic reticulum all over the sarcolemma, but specifically the zone of overlap, okay? Because this is where the magic happens, where we're going to get that interaction between the thick and the thin filaments. And in the video, you can see the myosin head, this little cocking head here, is physically pulling on the thin filament. And that's actually how muscle contractions work, okay? So what does the role that calcium plays in this coupling? Well, calcium binds to one of those proteins on the thin filament. If you remember, let me get my highlighter out here. Okay. It binds to the troponin. So calcium has a binding site on troponin, or I should say troponin has a binding site for calcium. Once calcium binds, where that troponin-tropomyosin complex was blocking the active site, if we can see now, when calcium binds, that troponin moves out of the way. So we can kind of see here on the animation, I know it's going kind of fast, but we can see the blue and the pink blobs kind of moving out of the way, allowing the thick filament to bind to the active site on those brown circles, which is our G-actin beach balls, okay? So once you have the troponin bound to the calcium, it moves, conformational change of that protein, moves the tropomyosin away from the active site, and now we can do what's called a cross bridge. It's the reaching out of the thick filament, the myosin head is binding to that thin filament on that active site of the G protein, the G-actin, sorry. And then when there's ATP, we're going to be able to do these power strokes, which grab onto the thin filament and moves everything towards the M line, okay? I know that was a lot, so make sure you can go through step by step. So we get excitation at the neuromuscular junction, that was in the previous slide. That excitation action potential runs down the T-tubules, causing the release of calcium to come out of the terminal cisternae. Calcium floods the zone of overlap, binds to the troponin, causes the tropomyosin to move out of the way. Once the tropomyosin moves out of the way, the active site is available. Myosin head reach up and grabs, and now we have a cross bridge formation. So this is all the coupling part, okay? So now what we're going to do is we're going to take a look at how that myosin head cycles through these repeated power strokes with being energized by ATP. Okay, so here's what we call the cross bridge cycle. So again, we're going to start at a resting cell. So step one, contraction cycle begins. So at rest, your troponin tropomyosin is covering the active sites. Your myosin head is kind of cocked back, like in this position, in the power stroke position. It's got an ADP and an inorganic phosphate. So an ATP molecule had come in, broken itself up, released the energy to cock that myosin head back into this position, okay? Then calcium ions come in, so here's our next step. Calcium ions come in, trigger the removal or the rotation of the tropomyosin away from the active sites. Once that happens, then the cross bridge can form. So then these cocked back myosin heads can reach up. It's kind of weird to do it in this position, this angle. So they're going to reach up and grab onto, tightly forming a bond to that thin filament, and then they're going to do a power stroke. And so that's this right here, the pivoting. The power stroke releases the ADP and PI, so that pops off, but it can't go back and recock itself until a new ATP molecule comes in. So that's this step right here. And when that happens, it lets go. The ATP comes in, it lets go, and then breaking that ATP to release the energy cocks that myosin head back, right? Myosin reactivation, and then we can start the cycle all over again. So as long as the active site is open, thanks to the calcium moving the troponin tropomyosin out of the way, as long as there's ATP to keep cocking the myosin head back and doing a power stroke and another ATP and power stroke and another ATP and the power stroke, as long as those ingredients are available, that situation is available, you're going to continue to move those thin filaments towards the M line. Okay? And so what we get, if you take a look at this sarcomere here, we can see the H band is pretty big. And then when we have contraction, we can see, that didn't really show that too well. Sorry, the H band is right here. And now the H band is that big. And then here we can see the Z disc is way over here, and now the Z disc is right here. So this is the physical shortening contraction of our sarcomere, end to end, all the sarcomeres are pulling on each other, which makes all the myofibrils get smaller. All the myofibrils are getting smaller. The muscle is getting smaller. The muscle cells are getting smaller. Fascicles, the whole muscle is getting smaller. All right, so this is what we call the cross bridge cycle. The animation here is showing you in that video. I'm going to post on the helpful links in Blackboard this animation. It's just a YouTube video, a good animation showing you all of these steps of the excitation, contraction, coupling, of the interaction between the thick and thin filament. So I know you can't see the full website here, but it's in the PowerPoint, but I also posted on the helpful links. So then just to review, so we have some overview slides of the contraction that we just went through. So we have the excitation at the neuromuscular junction. We have the action potential traveling along the sarcolemma down the T-tubules, causing the release of calcium out of the sarcoplasmic reticulum, flooding over the zone of overlap, okay, binding to the troponin, causing the tropomyosin to move out of the way, allowing the cross-bridge formation to form, and then powered by ATP, the contraction cycle is pulling the thin filaments towards the M line. All right, so that's our excitation, contraction, coupling kind of overview, real kind of in one shot. Now what happens when you're done? Now when you don't need to contract the muscle cell anymore. So you remember up at the neuromuscular junction, acetylcholine esterase removed the acetylcholine. So the signal stops. There we go. So here the signal stops because of ACHE, breaks down the acetylcholine. When we have no more action potential down the sarcolemma and the T-tubule, the cell actually burns ATP to move that calcium back into the sarcoplasmic reticulum. And it's active transport because we have to cram all of that calcium back into the high concentration area of the sarcoplasmic reticulum and the terminal cisternae. Once the calcium leaves the troponin tropomyosin, the active site is covered. Once the active site is covered, the cross bridge ends. You cannot bind the myosin head to active site if there's a troponin molecule or tropomyosin molecule in the way. And then your myosin head lets go, the contraction ends, and the muscle relaxes passively. So this is really important to remember. We do not actively lengthen the muscle cells. We can actively shorten them, but what if I want to lengthen, what if I want to straighten my arm? Do I actively push the bone away? No, my muscles are in antagonistic groups. So we have contraction here, but I have to rely on my tricep to straighten my arm. And when I contract my tricep, that's going to bring my bicep into resting length, okay? And so then if I'm contracting down here and I want to lengthen my tricep, I have to go like this. So it's kind of cool. Our muscles work in opposition, same like those body movements that we learned, right? So flexion and extension are powered by these antagonistic muscles. Just like abduction is going to be powered by this group of muscle, like your deltoids, and then your latissimus down here is going to reverse that and cause adduction, lengthening the fibers of my abduction muscles, okay? All right, so that is our video of excitation, contraction, coupling. So the excitation at the neuromuscular junction, the coupling of the calcium coming in and binding to the troponin, tropomyosin, moving out of the way, and then the contraction part of the cross bridge formation and the contraction cycle, the myosin heads pivoting, powered by ATP, and then relaxation as everything just kind of reverses itself, and the muscle lengthens passively either by an antagonistic muscle or gravity. Gravity can, pull your body parts back into resting position. Okay, so that ends that bit of physiology. We'll move on next to tension production in muscle fibers and whole muscle contraction. Okay, see you next time.